Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and IIT Guwahati. And welcome to the course Basics of Biology. And I'll be happy if you have any questions, and I'll be happy to discuss some of the topics from my own also. So kindly post your questions. This session is only for very short period of time. So good afternoon, everyone. If you have any questions, please post your question. Okay, so Rohini, what is your background? Rohini, if you could tell me the your background, I think I'll be able to help in terms of understanding the Latin much better. Because if you are a biology student, I will discuss accordingly. If you are a non-biology student, I'll discuss. Accordingly. Rohini, you are a biology student or non-biology student? Or biology student. So, okay, so platyhelminthes uh, is a uh, group where you have the flat worms. Okay, you know that the worms could be round, right? Because they have some mass inside, or they could be flat. Okay, so platyhelminthes means the helminthes which are plate-like. Okay, plate means they are flat, and that's why we have platyhelminthes. And classical example is Tinea solia or the and so and so platyhelminthes uh, a worm which is having a flat body. Then Sudarshan is asking why the chromosome organization in prokaryote is called nucleotide and not chromosome. No, uh, I think prokaryote, the genomic organization in prokaryote is not called as nucleotide. Uh, Nucleotide is the function, it's the subunit, or it is actually the unit which is responsible for formation of the DNA. Uh, Probably you have misspelled something. Okay. Um, but anyway, the genomic organization in prokaryote is also been called as sometimes the bacterial chromosome. But the bacterial chromosome is different from the eukaryotic chromosome because the eukaryotic chromosome, if you see very carefully, the chromosome is a very, very high level of organization. Okay. For example, when you have the DNA, this DNA is actually going to uh, uh, DNA is actually going to assembled with the histone proteins, and that's how they are actually the nucleus. And these nucleosomes, they are actually going to organize, or they will actually get the reorganized further to give you the 310 turns, and they will actually going to give you that these three turns are actually going to further uh, reorganized, and that's how you are actually going to have the very highly compact organization of the DNA in the form of chromosome. So we sometime by simplicity, or to make the things very simple, we call the genomic DNA or the genome of the of the prokaryotic system also as chromosome, but it is not the case. It is different from the eukaryotic, because eukaryotic chromosome is a very highly compact uh, organization of the DNA with the proteins. Uh, uh, Very good, Harini Narayanan. So I'm happy that you have also taken so this course is little bit more complicated, more of a higher standard than your age. I have probably made that it's very simple, so that should uh, then be able to follow the content. But in case you have not be able to understand something, you can ask me, and I'll try to see if I can make it as simple as possible. You should be able to understand. Roshan Jaha, you are asking what? How can a okay. Uh, what is your background, Roshan? What is your background, Roshan? Okay, so Roshan, you are currently a MSc student. So probably you are only two steps far away from going to be a professor in IIT. Okay, what you are supposed to do is just after your MSc, maybe you have to join any lab for PhD, and once you are going to be a PhD student, and you will successfully defend your thesis, and you will be a Dr. Roshan Jahan. Then, after that, you may actually pursue and for postdoctoral research outside. And depending on your expertise and experience, you can return back to India and you can apply to IIT as a faculty. And once IIT will select you, then your journey for becoming a professor in IIT will going to start. So when you so if you want to start your journey, what I will suggest is that should first choose the topics which you like, okay, and then try to be more dedicated and focused about your topics, and can actually be able to try to choose the topics which are in demand, and you should be able to be more comfortable in pursuing that particular type of research, and then try to come up with the publications, because ultimately what people are going to see is what kind of publications you have, what are the uh, what you have in your bag, because based on the publications in your PhD, somebody might give you a postdoc, right? So if you do a postdoc in a good lab, you will have the better publications, and based on these cumulative publications, probably there are many IIT which are uh, you know looking for the candidate, so they will be able to help you. Okay. Sudarshan, I have already prepared a presentation, so probably I'll discuss about the electron transport chain in due course. Uh, so, Roshan, I think if you want to be a professor in IIT, what I would suggest is you should be very, very dedicated, and you should take all the opportunities which actually going to fetch you a publication. Okay, so currently, like uh, regarding okay, so regarding the books for this particular course, since the course is being prepared for the non-biology students, although has the content for even biology student as well. Uh, you can actually start with the basic, very basic class 11 and 12 books. Okay, whatever you can get from the NCRT websites and all that. And once you are comfortable with those books and the content, then probably you can actually be able to explore even the higher standard books, which are the books which are, textbooks which people are referring in the class BSc first year or the and so on. I think I have given the list of books in my course portal as well, so you can actually be able to do it. Uh, so now, uh, uh, you can be able to apply to party. So I tracking the PhD psychology sessions, right? It's December, another thing in May, and you have already, you have started done your post graduation. uh, you can be able to apply. And he said, as I think you are asking, that there will be a gap, since for doing something else other than uh, so in IIT doing a PhD is an issue, and a year gap is not there, right? As long as you have the get qualified certificate or net qualified and all those kind of eligibility criteria, uh, IITs don't have any problem of the gap between your uh, graduation degree and PhD. Uh, so I think books. I already said about the books that we can refer the books. If you are a non-biology student, we can start with the class 11 and 12 books. If we have, if you are comfortable with that particular book and content, then you can actually be able to start with some higher standard books like from the BSc or even like the lineages and all those kind of high standard and books for getting the detail about the biomolecules, center of mouth, life, and all those. And for human physiology, what I have referred is more about like the content from the guidance and the other books. So, but I won't suggest you to you should follow those high standard books first. First, you start with the class 11 all books. Once you're comfortable with that and you know each and every detail about that particular book, then actually jump on to these books of the higher standard. The questions, I think regarding the questions in the exam, question is going to be of the multiple choice questions, and they are going to be, and yeah, it is going to be simple as like whatever we are asking, right? It's going to be very same. Okay, so now I'm going to share my screen, and I'll try to answer some of the questions which students have asked. Okay, so let me share my screen, and I will try to answer some of the all see my presentation. And okay, so first question. First question which I am asking is, I think some of the students were asking the about the linear plasmids. Okay, so I have asked a question in the one of the assignments that which of the following species have a linear plasmid. Okay, so this question is only the purpose of this question is that you all should be able to go through with the Google and other sources and try to search. Okay, so if you go with the Google and other searches, what you will find is that the linear plasmids are actually being present in many of the bacterial species. Okay, so what is linear plasmid? Linear plasmid is a linearized DNA when the DNA helix is cut in both strands at the same place, which means if you have a circular DNA like this, right? And if you cut at one point, what will happen is that it is going to open like this, right? So that's how it is actually going to linear. And uh, and so since linear plasticity, we have the differential migration and circular DNA, circular plastics. That's why you cannot use the same molecular methods to predict the size of the linear plasmid. So this is all about the linear plasmid, and I have kept some of these functions in the assignment simply because I want to ask, I want you to explore the uh, web as well as the content, but we could not discuss because the course has only 30 lectures. Okay. The course has only 30 hours to discuss. But many of the topics which are very interesting but cannot be discussed can be taken up like this so that if you are familiar with the DNA and all those kind of things, probably you can search the Google and try to figure out the. Now the next question, next topic is about the um, so next topic is about the electron transport chain. So one of the student is asking about the electron, explain the electron transport chain. So what is electron transport chain? Okay, so what will happen is that when you are actually having the biomolecules, whether it's glucose or fat, ultimately what we are generating the reducing equivalent, and this reducing equivalent are nothing but where you are actually taking up the electrons, you are extracting the electron glucose, and you are giving that electron to the NAD plus. Okay, so if you are making it giving the electron, remember that NAD has a positive charge, right? So when you are providing the electron, it is taking up that electron and getting converted into the NADH. Okay. So now, if you want, so this electron is the energy, right? This energy is being trapped into the NADH, right? Now, if I want to extract the energy in this, right, or if I want to extract the energy what is stored in this form, because the electrons are short-lived, so they cannot be, so they are actually short-lived, and they cannot be stored, right, for a very long time. So what you can do is you can just add this electron to NAD plus in a compound so that NADH is going to be generated, and NADH is a stable, it can store, you know. So after that, since we are always continuously looking for the energy, so what we can do is we can just convert the NADH into NAD plus, and that's how it is actually going to be electron. Now, this electron can be, so now you have extracted the energy, right? So if you have extracted the energy, or if you have extracted the electron, this is very high energy. Okay, so it has to be traveled throughout the different electron acceptors. Okay, and during this process, so you can see here, right? We have the NADH, right, which is being generated from the metabolic reactions, and that NADH is getting converted into NAD plus, and in that process, it has generated the electron. Okay. Now, electron is having the not only the charge but also contains the high energy. Okay, so it is actually a kind of a energy thing, right? So that electron is traveling from the complex number 1, 2, 3, and 4, and when it is traveling to 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 uh, so when it is traveling, it is actually uh, asking that particular complex to, when this complex is actually has to be taken up the electron, and when it will pass on to the next one, in this process it is actually going to transfer the proton from the mitochondrial matrix site to the inner membrane. Okay, so inner membrane space. So you are actually putting the lot of H plus. Okay, for example, this one, all other at every complex you are actually transferring the H plus, okay, onto this side. So in the inner membrane complex, you are actually accumulating the large quantity of H, and that H plus is actually going to give you the a potential. Okay, so that's why you are actually using a charge difference across the membrane. See, this is the this side of the membrane, this side of the membrane. The difference between these and this side is that you have a lot of positive charge, and this is side is going to be having the negative charge. And because of that, you are actually going to develop potential difference. And once you have the potential difference, the protons what are being generated into this electron transport chain is going to return back onto the matrix. But from where they can return? They can return only through a channel which is called as the ATP synthase. Okay, this is ATP synthase. And I'm sure when you have taken when you have seen the course, we have discussed about ATP synthase, how the alpha, beta, gamma, all those chains are present in the ATP synthase, and how the ATP, when the protons are coming out, how it is actually taking up the ADP and PI and joining them to give the ATP. Now, once you develop the ATP is going to be the currency of the energy, and that ATP is going to be used for the subsequent metabolic reactions, or is actually going to be run for the anabolic reactions and on so on. Okay? So this is the main purpose is that you are developing the chemicals, utilizing the energy from the glucose, fat, and all that, and you are withdrawing the electrons, and that's how you are converting the NADH. It is a chemical form of energy. Okay, so it's like as simple as like you are actually having the lithium ion batteries, right? You have seen that, right? You have a lithium ion battery, right? What is lithium ion battery is doing, right? When you are providing the current, it is storing the current. When there is no current, it is providing. So it is exactly the same, right? When there is no energy, the you are actually going to have the large quantity of NADH. So that is actually going to accept the electrons and convert the large quantity of NADH. And that is actually going to be a chemical form of energy, which will actually going to be go into the mitochondria, and then the mitochondria is going to extract that particular electron. And since that electron is very high energy, so it has to be traveled throughout the electron transport chain, and ultimately it is going to be received by the terminal electron acceptors, and is going to be disposed of. And in this process, a lot of proton is being expelled from the matrix side to the intermembrane space, and that proton will return back to the mitochondrial matrix to the ATP synthase, and that's how you are reducing the ATP. And that ATP is a readily form of energy which can be utilized into the metabolic reactions for the anabolic reactions, and you can use that for the synthesis of the proteins. You can actually be able to use that for synthesis of the nucleotides and so on. You can use that for the locomotion, which we are going to discuss when we are going to discuss modules also. If you have any questions, I'll be happy to answer about this also. And uh, what I will suggest or recommend is that this purpose of this live session is that you should discuss your queries related to the topics that we have discussed up to the module. Okay, rather than discussing about your future prospects, because I'm sure you aren't enrolled in any of higher universities or the higher institutions, and you are actually going to have which are very good in terms of which of explaining you about the how to become a professor or how to become a professor in IIT, because they are the best person to judge. They are the best to know what are your positives, what are your negatives, and so on. So suggest is that for all these kind of queries, better your teachers, because they know best how what your potential, what are the things you like, what are the things you don't like, and accordingly they can be able to do this. So I can give you my own example that when I was the BSc student, I went to one of my professors, uh, whatever the queries I was having at the time, and he suggested, and that's how I have pursued that pathway, and that's how I am here actually. So it's better to discuss with your teachers because they are the best to help you. Okay. Now, what is Chargaff's rule? Chargaff's rule is equivalent amount of pyrimidine in urate plus pyrimidine is actually going to give you a constant number. Means in a in a double helix DNA. Okay, only when it is double helix. Okay, not the single stranded. If it is single stranded, it's going to work. Okay, because then the Chargaff rule is not going to be applicable. So in a double helical DNA or double helical RNA, you are actually they are actually going to Chargaff rule, which means A is actually going to be equivalent to T and G is actually going to equivalent to the C, which means A plus G is equivalent to T plus C, for example. Okay. So how you can be able to use the Chargaff rules to actually know many things about the DNA. For example, if I give you the percentage of the A, for example, if I say you have a 20% adenine, okay, calculate the amount of C. You have to calculate the amount of C. So what I'll say is that A is equivalent to T, G is equivalent to C, which means if I have the 20% of A, I am going to have the 20% of T. Okay. And you know that A plus G is equal to T plus C. And this total is going to be 100. Okay. So if I put that into this, what I'll say is 20 plus G is equal to 20 plus C. And you know that the G is equivalent to C, right? So if I can write here is that 20 plus C is equal to 20 plus C. And together, this should be equivalent to 100, which means with the amount of C, C is actually going to be 30%. Okay, because if I put the 30% here, is going to be 20 plus 30 equal to 20 plus 30 because 50 on this side and 50 on this side. Okay, so that's how you can be able to use the Chargaff's rule to, to do this math. Okay, if you do this math, you can be able to predict whether the DNA or RNA is double helical or not, whether it is double or single. Okay, because if it does not follow the Chargaff rule, then it is actually going to be single stranded or it is actually going to be partially single stranded, which means it could be like this. In this area, it is going to follow the Chargaff rule. In this area, it may or may not follow the Chargaff rule because it could be a coincidence that the amount of A and T are actually equivalent. But if it is following the Chargaff rule, then that area or that region is going to be the double helix. Okay. Uh, I'm sure this is good enough to explain you about the Charga. If you have further questions, you can ask. Uh, I'm going to wind up this session in another 2 minutes. You're asking me how to develop a research interest. Research interest is a very difficult thing. If you cannot ask others to how. Okay. What you can do actually is you can actually be able to read the research articles, able to read the even the journal articles as well, journal science article, because that actually they are actually going to give you the interest. Okay, they will actually going to make the realization that the science is very very important and science is very interesting. For example, if you read about the dinosaurs, if you read about the biology and something, you will appreciate how well organized these organisms are being. Okay, and once you go through that, once you increase your base of your information, probably something will definitely come out from you, and it will actually going to help develop interest in that particular topic. For example, when I was a MSc student or when I was a master student, I was always having the curiosity, and I was having a local interest of how the cancer cells are spreading from one part of the body to another part, how the metastasis is happening, how the blood vessels are being formed, how the angiogenesis is working, and blah blah blah. And that's how I have started reading about those topics, and so that is actually a starting point for developing interest. Later on, depending on the opportunities, the situations, you might have to pick up the fresh topics and of course, uh, I'm sure this is good enough to discuss about this. If you have any more queries, I said, you should go to your supervisor, you should go to your seniors, to your teacher, and discuss in detail your problem. Discuss in detail about your queries, and they'll be happy to help you. So I think we don't have any more questions. I thank you all for joining this live sessions. And with this, I think I will be winding up the first live sessions. And we are soon going to meet with the second live sessions. And that time you can also ask support questions. So you can ask the questions of the module 1 to 4. You can ask the questions when to the subsequent module. Thank you.